Okay, 1 minute. Yes, yes, yes. Okay, okay. Yes, yes, yes. Happy Independence Day, everybody. So, there's one question out over here. In mothers suffering from HIV, colostrum won't be of good quality for the baby. Yes. So now what happens is in that also now there are different ways actually. Okay, now today we have all those kinds of medications now which are going to prevent the transfer of this thing from happening. So the virus from the mother to the fetus. Okay, also like for example, we have these drugs which are going to prevent even transplacental transfer of HIV. Okay, so we have got that kind of advancement as compared to what we used to have earlier. Yeah. Okay, chalo, chalo. Next thing. Okay, so guys, once again, we are talking about innate immunity, and now we need to see what all is going to be a part of innate immunity. Okay, like I told you in your textbook, it is given in a little different manner. They have combined local defense mechanism along with your actual innate immunity. Okay, so according to your textbook, guys, now innate immunity, this can be divided into your four main types. Okay, the first one, we have your physiological barriers. Okay, so we have physiological barriers. Second thing, we have anatomical barriers. Okay, third thing, we are going to have phagocytosis. Okay, and lastly, we are going to have inflammation. Okay, so we will see them slowly, slowly, one by one. What exactly is going to happen out over here? Hmm. Okay, chalo. So now first thing, guys, when we talk about your physiological barriers, now what are these exactly? See, these are going to be all your physical, your body features or body characters, which is going to prevent the germs from entering into your body. So for example, guys, what are you going to produce? What are the different kinds of fluids that you're going to produce? So guys, we have sweat. Okay, so sweat, then we have tears. Now, guys, first of all, when we talk about sweat, why aren't germs going to really grow in your sweat or something? Because it has huge amounts of lactic acid also. But at the same time, guys, if you keep the sweat as it is, meaning it stays on your shirt for a long time, then the sweat can be broken down. Because what happens, in your sweat, you're going to see little bit of urea being present, and germs can grow on urea. So, okay, then it will break down the urea to release your ammoniacal odor. But still, in the initial times, it is going to prevent the growth of your organism. Second, we have your tears. Now, guys, your tears, they have this enzyme called as lysozyme. And this lysozyme, it kills all the germs. And this is how immunity is provided to your eyes. Then we have nasal secretions. Especially in your nasal secretions, guys, we have something that is called as interferons. Okay, so in your nasal secretions and all, okay, uh, not necessarily only here, but there are other secretions also, like in your other bodies or in your other parts also, we have something that is called as interferons. Now, guys, what are these interferons? These are like signaling molecules. Okay, so for example, let's say if a cell is infected by a virus. Okay, so one cell is infected by a virus. Now, what it does is immediately it releases interferons. So, what is this? It releases interferons as your signaling molecules. Okay, these are going to act as signaling molecules. And what are they going to do? They are going to alert everybody besides that, oh, there has been a virus attack. There has been a virus attack. You keep yourselves ready to fight. So that way, okay, it acts as a signaling molecule and now alerts the neighboring cells. Okay, It alerts all the neighboring cells that are and makes them immune to that, immune to the virus. Apart from this, guys, we have other secretions also, like your HCl. Okay, for example, HCl, where is this? This is going to be present in our stomach. And now, because of the acidity, what is it going to do? It is going to kill the germs. Okay, fine. Along with that, guys, we have one more thing that is going to be body temperature. Now, what happens, guys? See, body temperature is your local defense mechanism. Now, for example, okay, if by any chance germs enter into your body, then what happens? Immediately, your body detects those germs and it starts increasing your body temperature. Now, why does it increase the body temperature? Because it feels that, oh, if the body temperature increases, the germs cannot survive the body temperature. They cannot survive the increased heat and the germs will die. But then in turn, what are you going to get, guys? You are going to get fever. But then, guys, fever is also your local defense mechanism. It is your body's mechanism by which it is going to kill the germs or make it really uncomfortable for the germs to grow. So that is all in your physiological barriers. Okay, but now let's try to see anatomical barriers. Now, guys, these anatomical barriers, these are the ones which are like going to be an actual barrier that we have talked about. So, for example, we are talking about skin, we talk about hair, we talk about mucus. Okay, we are talking about these things which are going to prevent the entry of germs into the body. Okay, these are the ones which prevent the entry of germs. Okay, next thing, guys, we have phagocytosis. So let's imagine. See, we had said that in your local defense mechanism, we are going to have WBCs, right? So this WBCs, they are going to show us phagocytosis. So this is going to be shown by WBCs. Once again, how is this really going to happen? Let's try to see this thing, guys. How is phagocytosis going to occur? So for example, guys, let's imagine we have a germ which is present out over here. Okay, so I'm just showing a transition kind of thing. Now, let's imagine the moment you have a germ out over here, what is going to happen? All the WBCs, they are going to be directed towards the germ, towards that area. How it is, we will see it in a while. Now, when you're going to see this thing, now as the germs are out over there, these WBCs have to squeeze out of the blood capillaries. So in the first case, they are going to come out like this. In the next case, they have completely squeezed out. Guys, what is this? This process is nothing but, yes, very nice, Devang, very nice. This is nothing but your diapedesis. What is diabetes? It is squeezing out of WBCs through your blood capillaries. Okay, so that is diabetes. But once the WBC is out, now what is it going to do? It is going to come and it is going to try to embrace that germ. Slowly, slowly, it is going to try to embrace it fully like this. Okay, and finally, it is now going to completely engulf this. Now, once it has got completely engulfed, then it is going to release certain substances, chemicals. We have your lysosomes who are going to act on this thing, and then they are going to kill the germ. Guys, what is this process? See, first is diapedesis, and the second is going to be phagocytosis. Now, guys, quickly tell me, which are the two WBCs in our body who are going to show us this process of phagocytosis? Quickly, guys, quickly. We've done this thing. Quickly. Come on, guys. Anybody? Lymphocytes? No. Basophils? No. Parnavi has given me one of the right answers. Ishita is right. Monocytes, yes. But I want others also. Guys, there are two. And leukocytes, right? Leukocytes. Guys, it is going to be monocytes and neutrophils. Leukocytes is nothing but WBCs. Those who are messaging me leukocyte is the other name for WBCs. So we have monocytes and neutrophils. Okay, both of them, they are going to show us this thing, guys. Okay, Next thing, we have inflammation. Now, guys, whatever was happening out over here, this is because of inflammation. Let's try to understand how. Okay, so let's imagine, guys, we have our, you know, a skin something like this. Okay, let's imagine a blood vessel is something like this. Now, what happens, guys? Let's say a mosquito has come and the mosquito has bitten you. Okay, so chalo, let's imagine this is our skin only. And let's say a mosquito has come and the mosquito has bitten you like this. Okay, zap, 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 zap. Okay, now what happens, guys? When the mosquito bites you, maybe the mosquito contains germs, the proboscis contains germs, and slowly you will see that the germs may be released out over here. Again, now it is not necessary that it has to be your malaria plasmodium only. No, 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 no. Okay, it can be any normal germ also. So what happens is, guys, at this place you are going to have germs. But because you have germs, guys, I need to take care of it. I need to kill those germs. Otherwise, what will happen? This will result into an infection. So immediately when the mosquito lives, what are you going to experience? You are going to experience swelling at that part. So slowly you're going to get a nice pimple. Okay, so you are going to have a pimple out over here, guys. But how is this pimple going to come out over here and what really happens? So, guys, what happens in this place is you are going to see that the basophils which are there, those are going to burst open. Tat, 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 tat. But basophils, guys, if you remember, basophils are responsible for the production of your histamines. Okay, so histamines are what, guys? Histamines are the ones which are going to bring about inflammation. Along with this, you're also going to see mast cells doing the same thing. Mast cells also release your histamines, because of which suddenly you're going to have inflammation. But now, the moment you have inflammation, guys, what is this inflammation going to result into? Inflammation will result into vasodilation, meaning you are going to have blood vessel increased in size in this region. Because there is increase in the size of the blood vessel, now you will see more blood reaching out over here. More blood reaches out over there, so you are going to see more WBCs are going to reach out over there through the blood. And now more WBCs immediately, once again, the same thing, first diapedesis and then phagocytosis. Okay, but in the meanwhile, guys, what is going to happen? How are you going to see? What are the things that are going to happen? So in that place where you're going to see inflammation, guys, first we are going to have swelling. Second thing, this is going to show you a redness. Why redness? Because more amount of blood is actually flowing out over there. Third thing, guys, if you check this thing, there is going to be a little bit of pain in that region. Okay, now why is redness out over there? It is going to be because of your vasodilation. And finally, guys, what are you going to say? If you actually check your pimples and all, guys, if you put your hand on them properly, you will feel that they are going to be a little on the hotter side as compared to the rest of the body. Okay, now once again, let me just, whatever I spoke of, let me just quickly put it down. So why we have going to have vasodilation? This is going to be because, okay, because of histamines. But who is the one who's going to release histamines? This is going to be released by your basophils and your mast cells, basophil and mast cells. And now what is going to happen? Along with this, they are also going to release other compounds like prostaglandins. Okay. So because of this, like all these compounds are going to be released. So what is going to happen? You are going to see more blood reaches that part. Okay, and now because more blood reaches that part, now you're going to see all these characters like swelling, redness, pain, vasodilation is going to be hotter. So you're going to see that because of this, once again you're going to have more WBCs, more WBCs. So you're going to see once again diapedesis plus your phagocytosis. And once this phagocytosis is done, guys, then ultimately what are you going to see, guys? Once the fight is over, then you are going to see pus formation. Now, pus is what, guys? you are going to see that pus is nothing but it is a mixture of either your plasma or your serum, okay, whatever you like to call it, okay, plasma serum, plus you are going to have dead WBCs, dead WBCs, plus your dead germs or pathogens, and plus you are going to have dead cells, okay, the dead tissue cells and all of them together, they are going to give you this creamy, off whitish or yellowish thing, which we call it as the pus. This is what we need to know with regards to your entire innate immunity. Guys, in your exams, if they ask you this, you have to write this thing only. You don't have to write local defense mechanism. Okay, you have to write this. Okay, and then once you have written this, guys, you also need to write down a little bit about this. What is this divided into specific and non-specific? Okay, everybody getting this part? In your exams, guys, in your boards, they ask you a short note on this thing for 3 marks. So you need to know this thing properly. Okay, so just have a look, guys. Just see if you are okay. Then we'll move ahead. One minute. I will just now just see, guys. Oh,
What did you say?
yeah. Go on, go on, go on. Hello, Arya. Are you asking something? I'm not able to hear you properly. Seems like she is unaware. She is. Just a moment. Okay. Okay, chalo. So everybody all right with this thing, guys? Fine. Let's try to move ahead. Okay. Chalo. Okay, now the next thing is, guys, what are the merits? Okay, now see, when we talked about your local defense mechanism, we had said that, local defense mechanism also has its merits, meaning it is going to start acting instantaneously. It is going to act over a wider range of things. It does not require any previous kind of exposure to any of the germs, right? That is what we had said. Same way, guys, your this thing in here, we are also going to have some merits of your innate immunity. So let's try to see what is it. Okay, so basically, if you look at this thing, guys, okay, what your textbook has done is they have mixed your merits of innate immunity and local defense mechanism into one thing. So the same thing we are going to put it down out over here. Okay, so first of all, guys, we need to remember always, okay, that this is going to be very specific. Okay, whenever we talk about this, we are going to have this as very specific. Now, why is it going to be very specific? Because you will always see that whenever we talk about your one antibody, okay, that is going to be like like one antibody will work on. Just a second, guys. One minute. Okay. So whenever we talk about your one antibody, guys, one antibody will work on only one antigen. It is not going to be like, one antibody will start working or fighting against your 10, 15 antigen. No, it doesn't happen that way. Okay, one antibody will work on one antigen only. Okay, so first of all, this is going to be very, very specific. Okay, fine. Second thing, guys, you are going to see that there are large number of antibodies produced, meaning huge amounts, diversity. Okay, so there is huge amounts of diversity of antibodies. Okay, third thing, guys, the most important thing, okay, which is out over here, this is that these antibodies, they can actually differentiate. Okay, antibodies can differentiate between what is self and what is non-self. Between self and non-self. And guys, this is one of the most important things. Has had it been, okay, had it been that it would not have been able to identify which is self antigen, which is foreign antigen, it would have started killing your own body cells and body antigens only. However, this is a not seen. Okay, so this is one most important characteristics. However, guys, sometimes by mistake you're going to see that things go wrong, and when things go wrong, guys, you will see your body cells being attacked by your own antibodies. And what are the diseases called? We call them as autoimmune disorders. We had done one of these things, guys, myasthenia gravis. Okay, so we had said that Amitabh Bachchan is suffering from myasthenia gravis, so that way. Okay, so that and last one, guys, last most important thing about your immune system as such. Okay, you are going to have immunological memory. Okay, now because you are having immunological memory, guys, today if you have fought with one germ, even after 20 years down the lane, you still have the ability to fight with the same germ because it has been registered in your mind, it has been registered in your body that yes, you have you have this entry done. So immediately next time, whenever that germ will come, first it is going to be checked that, oh, is he wanted? Is he one in the wanted list? If he is present in the wanted list, okay, you're just going to kill him right out over there itself. That is what is going to happen. So this is your immunological memory. Okay, but now see, we are talking about antibodies. But what are these antibodies? How are they? What really happens? Okay, so let's try to see what are antibodies and what are their properties. Okay, so now whenever we talk about antibodies, guys, antibodies, they are produced by lymphocytes. Okay, they are produced by lymphocytes. So we had said, guys, okay, the lymphocytes, they are going to be the smallest of the WBCs. Okay, but their nucleus is really large. Why? Because the cytoplasm is low. Okay, so we are talking about these. Now what happens, guys, is let's imagine there is a germ. Okay, so let's say the germ is having something like this. Okay, so he is going to have certain antigens which are going to be present on the top. Now I am just showing them as spikes, spikes, spikes. It can be of any types. Okay, so let's imagine these are the spikes, spikes, spikes. These are your antigen. Okay, now we also call it as epitope, but we will talk about this thing a little later. What is epitope and what is paratope? Paratope is going to be present on the antibodies. Okay, now what happens, guys? This lymphocyte, okay, remember I just told you this is going to be produced only by lymphocytes. This lymphocyte, it recognizes this germ. Okay, so this germ and the antigen which is going to be present on top. And now, in order to kill it or to detect it, what is this going to do, guys? Immediately, this produces these Y-shaped antibodies. Okay, so these Y-shaped antibodies. And now these antibodies, guys, they are going to come and they are going to attack onto this antigens. So they will fit like this. Now, once they fit like this, now you are going to have different mechanisms. Now, either these antibodies will help to release or help to bring about some reactions in this germ and break down its cell wall, or the moment we have this antibody, they are going to form a clump-like thing. So what you will see, you have macrophages. Okay, you have that is your monocytes or your neutrophils. Immediately, they are going to see out of here. Oh, there is this clumping thing. Immediately they are going to come out over here and they are going to show phagocytosis. This is how it is going to work. Two different ways in which these antibodies work. One, they are either going to help in killing the germ right out over there, or they are going to call out to your macrophages, and then the macrophages are going to come and they are going to show your phagocytosis this way. Okay, so this is how it is going to be. But now let's try to talk about what are the properties, what are these Y-shaped things that we are talking about. So guys, the first and the foremost thing is these are going to be your proteins. Okay, so when we talk about your antibodies, okay, antibodies we'll always write it as ABS, okay, AB or ABS. So antibodies, they are going to be proteins. And when I say they are proteins, they belong to a class. Okay. So in your bodies, we have different classes of proteins. Here they belong to a class of immunoglobulins. Okay. So immunoglobulins, immuno meaning your immune, globulins are the proteins. So proteins of your immune system. Okay, but now even if they are immunoglobulins, guys, we term immunoglobulins as Ig. Okay, this is how we write them. So they actually belong to 5 different classes of immunoglobulins. So what are the 5 different classes of globulins? IgG, IgM, IgA, IgD, IgE. These are the 5 different classes of immunoglobulins. Guys, you just need to remember this as GMADE. Okay, IG and GMADE. These, I mean, this is how I always keep it in mind. You can also keep it in mind like this, GMADE. Okay, but now, guys, when are these antibodies going to be produced? Are these antibodies being produced randomly and every now and then in your body? No. Guys, always remember these antibodies, they are going to be produced only and only when there is an exposure to an antigen. Unless and until there is no exposure, we cannot produce antibodies at all. Okay, so always remember these are produced on exposure to the antigen only. Okay, then the third thing. Okay, now how many varieties are we going to have? Guys, we sorry, we are going to have unlimited varieties of antibodies, guys. Okay, every now and then if you have a new germ coming up, For a new germ, you are going to have even like, sometimes you have 10, 15 antibodies targeting different parts of the germs. So you have unlimited varieties of your antibodies. Then next, like I said, guys, how are your antibodies? Your antibodies are going to be highly specific. Highly specific meaning they are going to be like, they can find out the particular germ only in a population of germs. So let's say there are so many germs out over there. Okay, and this is the one which is for which we have produced antibodies. So in this pool of this thing, in this pool of different germs, this antibody will not fight with anybody else कि अरे ये बाबा बाकी के germs are in between, so I will fight with them and then I will fight with them. No, you are going to see that this is going to come and it is going to fight only and only with this germ. This is called as being highly specific. Next thing, guys, another important property is your antibodies can recognize the antigen. Okay, antibodies have the ability to recognize the antigen. Now, how is this going to be, and what do I mean by this? See, guys, I told you that in the pool of these germs, this antibody is going to come and it is going to fit with only this. It is going to find only this one. How is this going to be possible, guys? Because this knows how this antigen is really going to look like. It knows how this germ actually is going to appear. That is why it is going to bind only with this and not with anybody else. So guys, always remember there is a slight difference between these 2 points, being highly specific and able to recognize. Okay, next thing. Okay. Now, some of the antibodies, guys, if you remember, some of the antibodies, they are going to be present right from the birth. Some antibodies are present right from birth. However, some, they are going to be developed in your life. Developed as you move on in life. Okay, fine. And next thing, okay, the next thing is, guys, how is the immunity going to be? So guys, the immunity that is provided by antibodies, it can be of two types. The immunity, it may be either short lasting or it may be long lasting. Okay, that depends upon the immunity that the antibodies that are going to be produced. So short lasting or it can be long lasting. That's it. That is what we need to know about your antibodies. Okay, so guys, if they ever ask you what are the properties of antibodies, how are you going to remember, guys? You remember this as PEUS rabies. Okay, just imagine you peed on the US and US got rabies. It doesn't make sense, but will help you to remember. Okay, so P, it is antibodies are proteins. When are they produced? They are produced only on exposure to antigens. How much is the variety? Unlimited variety. They are going to be highly specific. They are able to recognize. Some of them, they are present from the birth, and immunity may be either short lasting or long lasting. So PEUS rabies. Okay. In your exams, guys, they also ask you what are the different classes. Okay. So for 1 mark, they ask you this thing. IgG, IgM, IgA, IgD, IgE. Remember this as G made. Okay. Just see, guys, go through this. See if anybody has any doubts regarding this. Next thing that we're going to talk about is what are the different types of cells involved. Okay, this is very, very important. And for that, I would require your entire attention. Okay, everybody all right, guys? Uh, okay. So basically, acha, see what happens, see sometimes the immunity is like, you are going to produce antibodies. Okay. But these antibodies, now what happens, they may not last long, meaning, they are going to have a very short lifespan. So, even if the germs, they enter into your body, okay, let's say the germs have entered into your body, but antibodies are not there. The antibodies have just like, died off. Then what? You're not going to get immunity against it. Then you may start suffering from the disease. Sometimes what happens is some of the germs, they undergo mutations, rapid mutations. So in that case also, the antibody cannot find it, the antibody cannot detect it. Then the immunity is going to be short lasting. But for example, let's imagine we have chicken pox. Guys, I'm damn sure most of you guys must have suffered from chicken pox only once in your life. You don't suffer from it twice. Why? Because the moment you have suffered from it once, you are going to see that the antibodies and all, they are going to be present in your body for a really long time throughout your entire life. So no matter if the germ once again comes after 30 years or 40 years, still it is going to be detected as foreign and it is going to be killed that way. This is what is going to happen, guys. Okay, fine. Then someone is saying there is some mistake. Okay, what is the mistake? I don't get it. What mistake are you talking about? Can you please be a little? Is there a spelling mistake or something anywhere? I If there is a spelling mistake, it is like a slip of hand. Okay, so just understand it. Okay, guys, everybody all right with this thing? Fine, no issues. Okay, chalo. So guys, let's try to move ahead and let's try to see the next thing. Okay, now for this, I want your entire attention, guys. Okay, so please be there with me in this. Okay, and guys, in your textbooks and all, this has been given in the worst possible manner. Okay, it is really horrific. Horrific. Okay, so guys, the next thing we are actually going to talk about is cells of the immune system. Okay, so cells involved in immunity. Okay, so let's try to see who are we talking about and what really is going to happen. So guys, when we talk about your immune system or immunity as such, we said there are going to be two types of immunity. We have innate immunity and we have your acquired immunity. Once again, innate immunity, it is the immunity which is going to be present in your body right from the start of your life. Why? Once again, because of the virtue of the DNA. I also told you guys, innate immunity, this is also known as non-specific immunity. Okay, keep this thing in mind, guys. In your textbook, they have invariably used these kinds of terms. We said innate immunity can be divided into two, specific and non-specific. But remember, innate immunity generally refers to as non-specific immunity, and acquired immunity, this is generally referred to as specific immunity. Okay, guys, keep this thing in mind. In your textbook, suddenly they start talking about in between non-specific, specific. You will be confused what is happening. Okay, so this is the key to it. Okay, acquired immunity is also known as specific immunity, innate immunity, also known as non-specific immunity. Okay, now when we talk about the cells involved, okay, guys, we are talking about especially your acquired immunity. So let's try to see who are the cells involved in your acquired immunity. Okay, so guys, we are going to have two main cells out over here. Okay, so first thing, we are going to have lymphocytes. Okay, so we have lymphocytes, and the second ones that we have, we call them as antigen presenting cells or APCs. Okay, so antigen presenting cells, we call them as APCs. Okay, fine. So two cells majorly involved in your immune system, lymphocytes and APCs. Okay, but now first, let's try to focus our attention on lymphocytes. Okay, now guys, when we talk about lymphocytes, remember, lymphocytes are further going to be divided into two. How? Depending upon where they are going to mature. Okay, so we have two types of lymphocytes. We have B lymphocytes, and second, we have T lymphocytes. B lymphocytes and T lymphocytes. Now, guys, what is the difference between the two, B and T? There is a very minute difference. Guys, B lymphocytes are the one which are born in the bone marrow and they mature in the bone marrow. Okay, they are born in the bone marrow and they mature in the bone marrow itself. Okay, but now T lymphocytes, guys, now remember, T lymphocytes, these are the ones they are going to be born. Okay, remember, these are born in the bone marrow, but then they don't mature in the bone marrow. Rather, they're going to escape through the bone marrow and they move into the thymus. So let me just show you what is going to happen out over here. So guys, so we are going to see that we have these immature lymphocytes. Okay, immature lymphocytes present in the bone marrow. Okay, they are going to move to the thymus. Guys, we've already seen thymus in your endocrine system. So when we move into the thymus, guys, what is thymus going to do? Thymus produces thymosin. And immediately, what is this thymosin going to help into? It is now going to help in the maturation of your lymphocytes. Okay, so it produces thymosins, and thymosins, it helps to mature your lymphocytes. These lymphocytes which have matured, now what are these going to be, guys? These are known as T lymphocytes because they have matured in the thymus. Okay, everybody okay with this thing? Fine. But now let's try to see, guys. Now, whether it is the T lymphocytes or whether it is the B lymphocytes, okay? Always remember, both of them, they are going to be found in the blood. They are both of them, they are going to be seen in the lymph, okay? Lymph nodes, tonsils, everywhere. We are going to see them everywhere, lymph nodes. Okay, but now, once again, let's try to focus our attention on B lymphocytes. Now, guys, B lymphocytes, these are the ones who are mainly going to produce your antibodies. Now we are going to see how it is going to happen, but just for the timing, I'm just telling you guys. Okay, so we are going to produce, guys. There are different types of B lymphocytes, but for the timing, I'm just showing you this thing. So this produces your antibodies. Okay, and guys, the moment it produces antibodies, the immunity that you are going to get, okay, we call this immunity as humoral immunity. Okay, guys, once again in your textbook, they have invariably used this word humoral immunity, meaning the immunity produced or immunity given by your antibodies, or we also call this thing as antibody mediated immunity. Okay, so this is one thing. Okay, now when we talk about your T lymphocytes, now guys, T lymphocytes also, they are going to be of different types. Okay, now these T lymphocytes, they can be memory lymphocytes. Okay, they can be killer T cells also. So let's try to talk about killer T cells. These killer T cells, guys, they are going to kill the germs. Okay, directly, and that is why we call this thing as your cell mediated immunity. So they either kill the help in the killer T cells, and then they are going to trigger phagocytosis. Okay, they trigger phagocytosis. Okay, and because of this, guys, because we are making use of killer T cells, meaning some cells are involved in their killing, not your antibodies. That is why we call this kind of immunity as cell mediated immunity. Okay, guys, if you're going to use antibodies, then it is your antibody mediated or humoral immunity. If you're using cells to kill the germs, then this is going to be your cell mediated immunity. Guys, you need to know these two terms absolutely properly. In your textbooks, if you read it, sometimes they are going to use cell mediated immunity, humoral immunity. You will be confused, completely confused. Okay, and last thing, guys, where are we going to see both of these? So if we are looking at both, okay, we are going to see that both of them, they are going to travel in the blood, they are going to travel in the lymph, you're going to encounter them in the lymph nodes, okay, tonsils, Peyer's patches, okay, all your lymphatic organs and all, okay, tonsils, etc. So this is how your first thing, that is your lymphocytes, are going to be. Okay, guys, are you okay with this thing? Just have a look, everybody. Okay, main thing what you need to understand is why do we call them as T lymphocytes and why do we call them as B lymphocytes? T lymphocytes because they mature in the thymus, B lymphocytes because they mature in your bone marrow. Okay, just see if you're okay, then we'll move on to your antigen presenting cells, guys. These are like, a little different kinds of cells which are going to help bring about your phagocytosis. I will show you what really happens there. Is everybody okay? Sure, no issues at the moment. Only a few people are responding, guys. At least, respond. So I understand, like, whether you're understanding this or, guys, because as we move ahead, the chapter is slowly, I mean, this first half is going to be a little difficult. That is why. So we are going slow out over here so that we grasp all the concepts properly. Okay, chalo, let's move ahead. So now, guys, the next thing that we're going to talk about are your antigen presenting cells. Okay. Now, see, as the name suggests, antigen presenting cells, that means they are going to present the antigen somehow or the other. Okay, so let's try to understand how is it going to happen that way. Okay, so antigen presenting cells, guys, especially these are nothing but these are your phagocytic cells. Okay, so these are phagocytic cells. So they're going to show you phagocytosis, and by phagocytosis, they are going to do something. Okay, so for example, guys, who are we talking about? We are talking about monocytes. Okay, who are these monocytes? These are nothing but we call them as blood macrophages. Blood macrophages. Okay, second thing, guys, let's say we have histiocytes. Histiocytes, these are nothing but your tissue. Okay, in your tissues, we are going to have macrophages. So these are tissue macrophages. Then, guys, in your skins and all, okay, we have dendritic cells. Okay, we have dendritic cells, or we also call them as your Langerhans cells. Okay, this is generally seen in your skin, like I said. Okay, so for example, we have your Langerhans cells, and these are especially seen in your skin. Okay, but now let's try to see what exactly is going to happen, guys. Okay, so if you look at these Langerhans cells, okay, let me just show you. So the Langerhans cells, guys, they are going to appear something like this octopus kind of a thing, guys. Okay, something of this sort. Okay, this is how they generally appear, the dendritic cells. Okay, but now let's try to see, like, how it is really going to happen. And, like, how do these antigen presenting cells really work? Okay, sure. So let's try to learn a little bit about the same. Okay, so now guys, let's imagine we are going to have your antigen presenting cell somewhere out over here. Okay, so this is your antigen presenting cell. Now, the moment it realizes that, oh, there is this antigen out over here, this is the germ. Okay, now what is going to happen, guys? Immediately, this is going to start attacking the germ. So it is going to engulf the germ. Okay, so now let's imagine it has, in the next case, embrace the germ like this. In the third case, this is going to completely engulf the germ. Okay, now this is nothing but your phagocytosis. I told you these are nothing but your phagocytic cells. See, right in the first case, I have shown it is phagocytic cells. Okay, now what, guys? Now let's imagine you are completely eating this cell. Okay, the antigen is completely inside. Now, guys, what happens? The moment the antigen is inside, the cell is going to break it down. Okay, so let's say the cell is going to start putting in all the, in the enzymes and the acids and all, and then it is going to modify it. Now, what happens is, let's imagine the cell, this modifies the antigen. Okay, and now this modified antigen, whatever that is there, guys, this is now going to be presented on the cell. So let's imagine in the next case, guys, this is going to be like, it has completely eaten it, and now it has modified it in such a manner, guys. Okay, so it has modified it something like this. Okay, let's imagine in the next case, guys, this antigen, this is now going to be brought to the surface. So in the next case, you are going to see that this modified antigen, whatever that is there, guys, this is now going to be expressed, or now it is going to be shown on the top like this. Okay, so let's imagine you are going to see it something of this sort. Okay, so immediately this is going to be expressing it on the top. The moment this is expressed on the top of the cell, guys, now what? Immediately this is going to make all the cells, okay, this is going to be like, it is going to attract all the other cells that are present besides, and especially which cells it is going to express. This is going to attract your T helper cells. Okay, and these T helper cells, okay, so this activates, I say, because this is now expressed on the surface of the cell, this activates T helper cells. Now, what are these cells? We will just see it in a moment. Okay, it activates T helper cells, and the moment these T helper cells are activated, now what is going to happen, guys? This is going to attract all the other macrophages. Okay, so it attracts all the other macrophages. And now what? If all of them come out over here, now what are they going to know? That, oh, there is some problem out over here. Maybe there is an army of germs out over here, and then they are going to come and then they are going to just kill those germs out over here. This is how these antigen presenting cells work. Just a moment. Okay, so guys, this is how your antigen presenting cells, they are going to work. Everybody okay with this thing? Fine, no issues, guys. Okay, just have a look. Okay, so this is how the cells of your immune system are going to be like. Okay, just have a look at this thing. If everybody is okay, then we'll start moving on to the next part, guys. Okay, chalo. So guys, let's try to move ahead, everyone. Okay, chalo. So now, guys, the next thing that we need to know is, see, I told you, see, we are going to have these lymphocytes. Okay, so we have B lymphocytes, we have T lymphocytes, but both of them, they are going to work in a little different manner. So we need to see how are these going to work and what really happens. How are they going to be little different from one another? Okay, so now once again, guys, we are into your lymphocytes. Okay, and now in your lymphocytes, once again, guys, we are going to have two types of lymphocytes. So we have B lymphocytes and T lymphocytes. Okay, now what happens, guys? Let's try to see B lymphocytes. Now, first of all, when are these B lymphocytes going to be produced? Or what really happens? Okay, how are they going to start working? They are going to start working if they are going to be sensitized. If they are sensitized by the antigen, meaning if they detect the presence of antigen in their vicinity, or they are going to be sensitized by your helper T cells. Okay, helper T cells are some of the cells of the T lymphocytes. But then what do I mean by sensitized? Guys, sensitized means it is going to be activated or aggravated. Okay, sensitized means they are activated. Okay, that is the meaning of sensitized. In immunology, we always say, whenever there is like, any kind of action or any kind of something that is happening, we say it is because of sensitization. Okay, so always remember this thing. Okay, so B lymphocytes, they are going to be activated either by the antigen or by the helper T cells. Okay, but now let's try to understand what happens out over here. Okay. See, now let's imagine we have the B lymphocytes. Okay, the lymphocyte is something like this. Okay, now the moment the B lymphocytes, it is going to come in contact with the antigen. So let's say the antigen is something like this. Okay, so the germ, once again, we are going to have the germ, and let's say you are going to have this thing. Okay, now germs come in various shapes, various sizes. Okay, but let's say now, guys, the moment this one, okay, the moment this one sees it, okay, the moment this sees it, so let me just show you. Okay, if it sees this thing, guys, immediately what happens? You are going to see that this lymphocyte, it immediately produces clones of itself. It produces clones, meaning exact copies of this thing, but then both of them, they are going to be different. Now the moment it produces clones, it starts giving rise to two different types. Now what are the two different clones it produces? So the first one, guys, it produces is memory B cell. Okay, so memory B cell, this is going to keep a memory of like who this antigen is, what are the characteristics, what are the features of this antigen. So those cells, we call them as memory B cells. Okay, and the second one, the second type it is going to produce is, it is going to produce your plasma B cells. Okay, now remember, the plasma B cells are the one who produce your antibodies. Okay, so memory B cells and the plasma B cells. Okay. Okay, so now, okay, so just understand what has happened. Okay, so the moment this lymphocyte it sees the antigen, the moment it sees or it comes in contact with the antigen, it is going to produce clones of itself. There are two types of clones. We have memory B cells and we have plasma B cells. Now, what is the memory B cell going to do? Memory B cell, it keeps immunological memory. Okay, meaning this is going to keep it in your mind that for generations and generations that that this is generation meaning for years and years, decades and decades, that this is the germ which is not wanted, unwanted in your body. And plasma B cells, like I said, these are the ones which are going to produce antibodies. Okay, it produces antibodies. But now when it produces antibodies, guys, there are two types of antibody. I mean, two ways in this which the antibodies can move. Now, either they may be free floating. They are going to be floating freely through your blood and also that is free floating. Or the second one, it can also be attached to certain cells of the body. So two ways these antibodies can be seen in your body, attached to certain cells. That's it. This is the first thing that we need to know. Okay. Now we have something like how these antibodies are going to work and fight with the germs, that we will see it. But first, let's try to move on to your this thing. Okay, onto your lymphocytes, T lymphocytes, and then we will see how it is going to work exactly. Okay. So now when we talk about T lymphocytes, how are these T lymphocytes activated? So these T lymphocytes, guys, they are sensitized by the antigen. Okay, only by the antigen. Okay, they are sensitized by the antigen. And now, just like how this B lymphocyte had produced clones, the moment T lymphocytes are, they are going to be activated by the antigen, they also produce clones. But now, these clones, guys, these are of four different types. So let's try to see what are the four different types of clones. So the first type of clone is, guys, this is known as the helper T cell or the T helper cells. Okay, helper T cells, or we also call this thing as the T helper cells. Okay, whatever you like to call this is fine. Okay, but now these helper T cells, what are they going to do? Guys, the moment we have these helper T cells, they are going to produce something. What are they going to produce? They produce lymphokines. Okay, very, very important. They produce lymphokines. Okay. But now, the moment they produce lymphokines, guys, what is the job of this lymphokines? What is this going to do? So immediately you are going to see that these lymphokines, first, they are going to bring about proliferation of T cells. Okay, so they are going to bring about proliferation of T cells. First thing. Second thing, guys, they are going to sensitize B cells. See, I had told you out over here, see, they can, the B cells can be sensitized by your helper T cells. So see, that is the job. And the third thing that is this is going to do, guys, this is also going to attract macrophages. It attracts macrophages. That's it. That is the job of your helper T cells first. Okay, now guys, this is one type of your clone. Second thing, second type of clone that is going to be produced, that is called as your killer T cell. Okay, killer T cell, or we also call this thing as cytotoxic T cell. Okay, now as the name suggests, okay, killer T cell or cytotoxic T cells, definitely this is going to bring about the killing of other cells, right? So now what is this going to do, guys? This killer T cell, this directly attacks and kills the germs. Okay, directly attacks and kills germs. But how does it achieve this thing? Guys, this is achieved by the production of another compound by, we call this thing as perforins. Guys, very, very important. So see, first helper T cells, lymphokines, second cytotoxic T cells, perforins. Now, guys, what are these perforins going to do? Perforins actually bore holes into the germ's body. It bores holes into the germ's body. And now, once that is done, kachak, it is going to kill the germs. Okay, and it is going to send the toxic substances, sends toxic substances, and finally it kills the germ that way. So this is going to be your cytotoxic T cells. But now see, guys, because see, earlier also we said that, T lymphocytes, I mean T lymphocytes, they are going to show you cell mediated immunity. See, this is the reason for it. It has this killer T cell. The killer T cells are cells which bring about the death of other cells. So that is why we call this thing as cell mediated immunity. Okay, fine. So now the next thing, again, the next thing we are going to see, the third thing. The third thing are called as your suppressor T cells. Okay, now what are these suppressor T cells? Now, guys, see, I told you your antibodies or your cells should not fight against your body, right? So there has to be some kind of mechanism, right, by which we are going to identify your cells and the foreign cells. That is done by these suppressor T cells. Guys, suppressor T cells, their job is to suppress the immune system. So this suppresses immune system, and because it suppresses the immune system, it helps to identify who is self and non-self. Okay, helps to identify self and non-self. And by doing this thing, okay, by doing this, it does not attack your own body. That is the idea. And finally, guys, the last kind of thing, the last kind of your cell that it is going to produce, we also call this thing as memory T cell. Now, memory T cell, guys, this is nothing but, once again, this is going to keep your immunological memory. Immunological memory. That's it. These are the few different cells of your immune system, guys. Okay. Now, still, we haven't completed this thing, guys. We still need to see how our antibodies going to work. Okay. We have just talked about only the cells. First, guys, just go through the cells, see if you have any doubts, anything you wish to ask. Yes. Guys, remember, this part has been given in the worst possible manner in your textbook. Okay. So keep this thing in mind, guys. What is the difference between memory B cells and memory T cells? Nothing. Only difference is their point of origin. Memory B cells and memory T cells, both of them, they show the same thing, immunological memory only. Only difference, memory B cells, they have matured in the bone marrow, and memory T cells have matured in the thymus. That's it. That is the only difference. Okay, anybody else? Guys, have you understood these things? Okay, if you want, I will just once again give you how the flow is, what we are doing. So guys, first see, once again, what we did first, we started talking about how is the immune system. Then we started talking about what are the cells involved. So in cells, we talked about lymphocytes and antigen presenting cells. In lymphocytes, we have B lymphocytes and T lymphocytes, but B lymphocytes and T lymphocytes have various types. So see, we are seeing the types of lymphocytes. Like, we have B lymphocytes, then in B lymphocytes, memory B cells, plasma B cells. T lymphocytes, helper T cells, killer T cells, your suppressor T cells, and memory T cells. And here we have antigen presenting cells. This is how the flow is, guys. Okay, everybody getting this part right? Sure. Okay, let's try to move ahead, guys, and let's try to see the next thing. Okay, the next thing is how are these antibodies going to work? Because see, in your memory B cells, memory T cells, sorry, in your here memory B cells and plasma B cells, we said plasma B cells are the ones who produce your antibodies, right? So now, how are these antibodies going to work? That is a challenge. So let's try to see how is it really going to happen. Okay, so antibodies, when we look at them, guys, antibodies work in three different ways. Okay, in three different ways. But then, how was it? Let's figure this thing out. Okay, so the first method, we call this thing as agglutination method. Okay, agglutination meaning, guys, this is nothing but clumping. Okay, so if you want in brackets, I'll just put down this is nothing but clumping. So you're going to see clumping form. Okay, let's try to understand how is this going to happen. Okay, so guys, let's assume this is your germ. Okay, that was present out over here. Okay, so this is your germ. This is another germ. Okay, something like this. Okay, let's say this is another germ. Okay, something of this sort. Guys, now what happens, when we talk about these germs. Okay, so let me just once again copy them. Now, the moment these germs are going to be present, guys, what is your body going to do? What is your B cells going to do? That is your plasma B cells. Immediately, they are going to start producing antibodies. Now, these antibodies, they are going to be directed towards these. In the next case, guys, what are we going to see? We are going to see that, oh, these antibodies, they are going to come and now they are going to form a complex kind of a thing, guys, out over here. Meaning they are going to just act as if, all of them, they have like clumped together like this. Okay, now when we have a situation like this, guys, what is this going to do? Like this clumping thing, okay, immediately this attracts macrophages. It attracts macrophages. And now, once again, macrophages meaning who are we talking about? We are talking about your monocytes and your neutrophils. Okay, both of them, they are going to come, they are going to start eating them away because this is a signal for them that, oh, here there is clumping or here there is agglutination. Oh, meaning something is wrong. It is time for them to act. So that way, so they are going to come out over here and immediately they are going to show us phagocytosis. This is nothing but your agglutination method. Okay, but now on the similar lines, guys, we have the second one that is called as opsonization. Okay, opsonization. Now, guys, in opsonization, let me just tell you, you are going to see that, both of them, they are almost on the same lines, but a little different, a little different. Let's say in your opsonization, guys, you have one germ. Okay, so this is your germ. Let's say something like this. Now, here only one single antibody is quite capable enough of bringing about the destruction of this germ. So I'll show you how. Okay, so now guys, you will see that the moment this germ is out over there, you are going to produce antibody. This antibody is going to come and it is going to fit onto the antigen. Always remember the antigen antibody is a very specific reaction. Okay, but now guys, the moment it is going to be like this, now what happens? It starts sending signals, meaning like, this is like a beacon kind of a thing that, ting, 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 it is going to flash, 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 and it is now going to attract the attention of macrophages. So see, because this has happened, immediately this sends signals to macrophages. And now these macrophages, they are going to come and they will show you phagocytosis. That's it. This process is your opsonization process. Okay, next. Okay, the next one, guys, we are going to have the third thing. The third thing is nothing but we say this is your neutralization. Okay, but neutralization of what actually? Neutralization of toxins. So, guys, for your toxins, we are going to produce antitoxin. So, toxin, antitoxin, they are going to neutralize one another. That's it. And after this, guys, so basically, what is the example for of this neutralization reaction? So, we are going to have toxin for your tetanus. So, we produce tetanus, anti-tetanus toxin, and for diphtheria, we have the other one, diphtheria, that way. Okay, so this is nothing but neutralization. So neutralization of your germs, uh, your toxin. Always remember, guys, how is this neutral? If you want, I just mentioned because okay, so we are going to see toxin and antitoxin. Antitoxin is nothing but this is produced by your WBCs, right? Okay. Chalo. So guys, this is how we are going to have the different cells of your immune system, and this is how it is really going to occur. All right, everybody fine with this thing? Okay. Next thing that we are going to see is, see guys, we've been talking about like, yeah, antibodies, antibodies, antibodies. Next thing we're going to see is what are these antibodies really like? What really happens out over there? Okay, so that is what we will see. Okay, but guys, what we will do is we will break over here for 10 minutes. Okay, so after 10 minutes, once again, we will continue. Okay, so it is reaching like, 1 minute. It is, I believe it is 8 48 or something, 8:48, 8:49. Okay, so guys, 10 minutes break. So 9 o'clock we'll be back again. Okay, and then we'll continue for the last hour. Okay, so 10 minutes break, guys. It is okay. Okay, 10 minutes break and then we'll come back. Yeah. Okay, chalo, let's start, guys. Everyone. Okay, are you guys back? Guys, kindly message if you are back. Yeah. Okay. Chalo. So now, guys, let's try to move ahead and let's try to see. See, I told you we are talking about your antibodies. Okay, but now let's try to see what are your antibodies going to be like. Like, how do they really look? What really happens out over there? So let's try to see how are your antibodies and what really happens out over there. Okay. Okay. So the next thing we are going to talk about is the structure. Okay, so now guys, we said, when we talked about the structure of antibodies, we said the antibodies, they are going to be Y-shaped like this. Okay, so they are all going to be this way. But now what we need to do is, guys, we need to zoom in onto them and we need to see what are they actually made up of. Okay, so guys, what we see is, see, these Ys that we are drawing, actually, they consist of 4 polypeptide chains. So how is it? Let me just show you. So see, this is one chain which is going to be on the inner side. This is another chain which is going to be on the inner side, like this. Okay, these are first two inner chains. And now you are going to see that outside, you are also going to see one more chain like this. Okay, when I say chain, what are these chains? Chains means it is nothing but your polypeptide chains. Okay, so polypeptide meaning once again your protein chains like that. So you're going to see it this way, guys. Okay, so this is how that entire thing is going to look like, the your entire antibody. But now, guys, what are we going to see is, okay, let me just, okay, anyways, chalo. Okay, and what are you going to see, guys? These two polypeptide chains, whatever that are there. Oh, just a moment, someone's at the door. Okay, so now, guys, we are going to have these two polypeptide chains, and see these two polypeptide chains, we need to hold them together. So what we will see is in between, you are going to see some disulfide linkages. So ये color दिख नहीं रहा है. Okay, so you will see some disulfide linkages, SSSS. Okay, that is disulfide linkage. So out over here, same thing goes out over here as well. So like this, guys. Okay, same thing out over here. In between these two also, you are going to see disulfide linkages. Okay, now, okay, let me just mention these are all disulfide linkages. Okay, now what are these chains that we are showing, guys? Okay, let's understand this thing. See, the ones which are going to be on the outer side. Okay, we say these are going to be light chains. Okay, these are your light chains. So light chain, this is your light chain. Okay, and the ones which are going to be on the inside, these are known as your heavy chains. Okay, so if you want, okay, this heavy, this one is also heavy chains. So these are heavy chains. Now, when we talk about these things, okay, what you will also see, guys, is at this point, see this point, we call this thing as the hinge, where there is this bend present. So this region, we call this thing as the hinge region. So that bend region is basically your hinge. So this is how your actual, antibody is really going to look like. Okay, but now still we have to see something more. Let's zoom in into this thing a bit and let's try to see some parts of this thing. Okay, so okay. So now once again, drawing the same antibody, guys. Okay, so you are going to have structure like this. Okay, so once again, some part like this, some part like this. Okay, now what happens, guys? Here, there are going to be a few regions which are going to be common to both, some regions which are going to be uncommon to both. Okay, so let's try to figure this thing out. So now, guys, what happens, in both of these things, you are going to see that there are going to be this portion. This portion of both the chains, it is going to be common to one another. Okay, so it is going to be common to one another like this. So this chain, I mean, this thing, uh, let me just, so, okay, common to one another, meaning this is the one, this region is the one which is going to be common to your antigen that you're going to be looking at. Okay, so this is going to bind with the antigen. So we call this region, okay, this top portion, whatever that is there, we call this thing as the FAB region. Okay, FAB, or we also call this thing as the variable region. FAB or the variable region. This is the one which is going to bind with the antigen. So let me just show you on the top. Let's say this is the place where it is going to bind with the antigen. This point, okay, where it is going to bind with the antigen, we call this thing as paratope. Okay, and the paratope is going to fit with the epitope of the antigen. So let me just show you. So this is the antigen. Okay, let's imagine the antigen is like this once again. So this is actually the epitope of the antigen. Okay, so this paratope, epitope, they fit into one another. Okay, and now guys, you will see that every antibody which is present, every antibody will have a different, this part, the upper part. See, whatever this we are showing, this region, this is going to be different for everybody. Okay, it is different for everybody. But all the antibodies will show a common region. See, from here until here, everything is going to be common. The structure remains the same. The composition remains the same. So we call this region as the FC region or the constant region. The constant region. This way. Okay, and now guys, most of the times, because this entire thing is going to be Y-shaped structure, you are going to see that every antibody has two places where it can bind with the epitope. As there are two binding sites, okay, now remember, as there are two binding sites, we call this thing as bivalent antibody. This is known as bivalent antibody. Okay, everybody all right with this thing? Sure, no issues, guys. Okay, just have a look. This is the structure of your antibody. And guys, in your exam, they will ask you to draw this thing for 2 marks, or if you're lucky, 3 marks. You have to draw both these structures. One where you're showing heavy chain, light chain, and second where you're going to show FAB and the FC regions. Okay, but now guys, I will give you a way. Now what happens is, in your exams they will always ask you epitope is present on what and paratope is present on what, and you will get confused. Okay, always. So guys, remember this thing as parab. Okay, just remember this as array parab. Okay, parab is paratope antibody. So what remains is going to be your epitope on your antigen. Okay, just remember this thing as parab. Okay, fine. But now, guys, let's try to see how is this antigen antibody reaction going to take place. So we've always said antigen is going to react with antibody, and then I mean, both of them, they're going to be like going to be clumping together and all. So let's try to see how does this thing work. Antigen antibody complex. Okay, so now once again, guys, let's imagine this is your antibody. Okay, and now this has to this has to react with your antigen. Okay, so now let's see how it is going to happen. So guys, out over here on both these things, we said we are going to have the paratope. Okay, because it is going to be your antibody, we have paratope. And now the epitope basically is going to be present out over here. So let's imagine your antigen is present like this out over here. Now, when both of them, they are going to come in close contact, you are going to see that the antigen is going to fit out over here properly. So in the next case, we are going to see, and plus because this is a bivalent thing, you are now going to see that this antibody is bonded to your antigen like this. Like this. This is whatever that has happened out over here, guys. This is nothing but your antigen antibody complex. Antigen is written as AG, antigen antibody complex. This is what we need to know. Okay, so guys, just once again go through this, just see if you're okay with the structures of both. If you have any doubts, then ask, or else we will move on to the next part, that is nothing but your blood grouping. So when we talked about your circulatory system, I told you, blood grouping and all, will do it in your human health and diseases. So next part is nothing but your blood grouping and all the different things associated with it. Okay, but first just see if everybody is okay with this. Okay, everybody all right? Okay, guys, everybody okay? Have you guys understood this part? Okay, so until this part, guys, it was a little something new, something which you had not known of. Okay, but now as we move ahead, now once again things are going to be really, really simple. Okay, guys, everybody okay with this? Kindly reply quickly, quickly, quickly reply, guys. Okay. Okay, now the next thing, guys, next thing is very, very simple. Okay, half part of it we had already seen this thing in your circulatory system, but now we'll do it once again in proper detail. Okay, so guys, what are we looking at? We are looking at your ABO blood grouping system. Okay, so ABO blood groups. Okay, so now first thing, guys, who were the people who were involved in doing this blood group or finding out what are the different varied blood groups? So the person who was involved, his name is Carl Landsteiner, and he found this thing out in the year 1900. Okay, so we have Carl Landsteiner. Okay, now what was he doing? So Carl Landsteiner, he was working on the blood groups, and he found out that there are three blood groups: A, B, and O. Okay, at that time he had just found out three blood groups: A, B, and O. But now what happened was, a couple of years later, that is 1902, his students, okay, Carl Landsteiner's students, D. Castello and Sterling. Okay, D. Castello and Sterling, they found out the fourth blood group, that is AB. So in all, now we are going to have four different types of blood groups: A, B, AB, and O. Okay, but now once again, how is this really going to be, and what are the different blood groups? Let's try to see. So guys, we have, let's imagine we have blood groups, and how are these going to be? And let's just try to figure this thing out. Okay, so let's say this is your RBC. Now everything is going to be based on the RBC, okay? Like how the RBC, what antigen is going to be present. So I've also told you, like, these antigens, they are going to be because of the various carbohydrates that are going to be present. Okay, so it is because of that. So let's imagine this. So let's say this is your RBC, and let me just quickly copy this and paste it. Okay, so guys, these are the four different types of RBCs. Now, how are they going to be? Let's try to see. So for example, let's say on the first RBC, you are going to have an antigen, okay? An antigen which appears to be something like a red dot. We say this is a antigen, okay? Now, if it is, and let me just draw it out over here also, because this thing. So if there is antigen present on top of this thing, guys, this A antigen, we call this thing as A blood group. Okay, so this becomes your blood group A. Okay, likewise, we have another antigen which is going to be present, which is B. Let's say it is blue and dot like this. So if it is going to be this way, guys, we see this is going to be B blood group. If we are having both the antigens present, A antigen also and B antigen also, then we call this thing as the AB blood group. And if for any reason we do not have any antigen on the top, then guys, what is going to happen? We call this thing as O blood group. And depending upon this, guys, see, we are having four different types of blood groups. Now, guys, for the time being, I'm not talking about what is positive, negative, AB positive, AB negative, A negative, A positive. We're not talking about that. We are just talking about plain ABO blood group system. Okay, when we talk about positive, negative, we are adding RH system into it. Okay, but now let's try to understand a few things, guys. Okay, so guys, when we talk about your blood groups, okay, now what happens in your blood groups? Every blood group is going to be determined by some gene. Okay, so who is the gene which is responsible for your blood groups? We have said it is going to be gene I. Okay, now gene I can be of two types. Either it can be I or it can be small I. Now, in this chapter, what we are going to do is instead of writing small I, we are going to write this thing as IO. So actually, there are going to be three genes. Okay, let's try to see. We have IA for your A antigen, IB for your B antigen, and let's say IO for no antigen on the top. Take it this way. So this is how we will put it down. Okay, fine. Chalo. So now let's try to see on the basis of this. Okay, let me just put it out over here so that we can have a look. Okay, so now guys, we are talking about blood groups. So let's try to see how are the blood groups and some other features of it. So we have blood group. Second thing that we're going to talk about is what is the genotype, meaning what is the gene thing, okay, genotype. Third thing, we are going to see what is the antigen present on top. And fourth thing is what is the antibody that are going to be produced. Okay, fine. Let's try to see. Okay, and always remember, guys, keep this thing in your mind. Okay, keep this diagram in your mind. If you keep this thing in mind, then I'm damn sure you're never going to go wrong. But if you miss this diagram, then guys, and if you try to just remember it on your own, try to mug it, then 101% you are going to go wrong, guys. Okay, so just keep this thing in mind. Okay, so now first thing, let's try to see. So we are talking about your blood group A. Now, blood group A, guys, there can be two genotypes. So how have we said? So once again, I'll just say we have, sorry, IA, okay, and IB. If you remember, we said both of them, they're going to be dominant over small I or I0. And individually, you're going to see that IA is going to be almost co-dominant, okay, with your IB. This is how we had said, if you remember in genetics, okay. But now let's try to use the same thing out over here. So if you look at the genotypes, what is the genotype going to be? So either you can be IA. So this is going to be A only, or if you are heterozygous, then you can be IAIO. This is the other thing. Okay, now see, what is the antigen present on the top, guys? See, the antigen present is antigen A. And what is the antibody produced? Now, guys, under normal circumstances, are you going to produce antibodies against any other blood group? The answer is no. So, guys, what you need to understand out over here is antibody is going to be produced only when your blood comes in contact with others' blood. Okay, only when another blood group, okay, is injected into your body or your blood comes in contact with another blood group. Only when your blood comes in contact with other blood group. Okay, unless and until this does not happen, guys, your blood will never produce antibodies against anything. This is the most important thing, guys. Okay, but now antibodies is going to be produced against what? So see, here we have only B antigens, two antigens, A antigen, B antigen. So here, if by any chance, let's say I take this B blood group and I put it into your A blood group, then B blood group will be foreign for A blood group, right? So what will it do? Immediately it will start producing antibodies against only the B blood group. Okay. So what is the antibody produced? The antibody produced, we will say this is going to be antibody B or anti-B. First thing, guys, have you understood this first line? Just tell me yes or no quickly. Quickly. Most important thing is when is this antibody going to be produced, because most of the times nobody tells you like why will this antibody be produced, and then you're like completely confused. Okay, now if you have understood this thing, guys, then on the same lines let's try to see the second one. Okay, the second one is blood group B. So what is the genotype going to be? So you are going to see either it is going to be IB, which is homozygous, or it is going to be IBIO, this way. Now, what is the antigen present on the top? See, this blood group antigen present is antigen B. But now what is the antibody? Now see, let's imagine you take A blood group, you poke it into your B blood group. For B, A is going to be foreign. So what will it produce? Antibodies against A blood group. So here only and only when the A blood group comes in contact with B blood group, the B blood group will produce antibodies against A. So this is antibody A. Okay. Third thing, let's try to see the third one. Okay, now the third blood group, see, we are going to have AB, right? So what is the genotype? Genotype is going to be only one, where both of them, they are going to be co-dominant. So IA, IB. And now what is the antigen present? Both A also and B. But now, are they going to produce antibodies? See, you take A blood group, you put it out over here, it is going to be recognized as self. You take B blood group, you put it out over here, it is going to be recognized as self. You take O blood group, you put it out over here. O blood group has no antigen, so it is not going to initiate any reaction. So here, is A blood group going to produce any kind of antibodies against anybody? Yes, guys, what do you think? Is it going to produce? Quickly answer. Is A B going to produce antibodies? No. Very nice. Okay, so here there are going to be no antibodies. Okay, but now on the last side, okay, just see out over here, guys. Lastly, we are going to have O blood group. On O, is there any antigen present? No, there is no antigen present. Okay, so first we have genotype. What is the genotype? It is going to be IO. What is the antigen? There is no antigen present. And guys, if you take any of the blood groups and put it into O, it will immediately start producing antibodies. So what are the antibodies that we are going to see? Here you're going to see both antibody A as well as antibody B. Antibody A as well as antibody B. Guys, this is what is going to happen. Okay, so first just see if everybody is okay with this chart, guys, and then we will see once again who is going to donate blood to whom. So the blood grouping chart. Okay, everybody all right? Guys, everybody all right with this part quickly? Okay. Chalo. So let's move ahead, guys. Okay, so now the next thing that we're going to see, okay, once again, you have to keep this thing in your mind only. Okay, so these, let me just copy this. Okay, chalo. So now the next thing is, guys, we are going to see your blood transfusion, or who can donate blood to whom, who can accept blood from whom. So let's try to see, guys. Okay, so for that, once again, see, let us keep it this way. Okay, so now guys, we are going to see who is going to be the donor and who is going to be the acceptor. Okay, so let's say we have this thing. Okay, guys, I'm drawing this this way. Okay, so first we are going to have acceptors on this side. Okay, so these are your acceptors. Okay, and the ones which I'm going to be listing down, those are going to be your donors. Okay, so now let's try to see who is your donor. Once again, the same thing: A, B, AB, and O. Okay, so this and these are going to be your acceptors. Let's try to see. Okay, now guys, let's try to understand. Oh, just a moment. Okay, so now let's try to see who can donate blood to whom and who can receive blood from whom. Okay, so let me, so, okay. So now let's say, can A donate blood to A? Okay, so let's see, we are seeing this way. Okay, can A donate blood to A? Yes. Okay, so let's try to see it from this angle. Can A donate blood to A? Yes. Okay, because both the antigens are the same, so not an issue. Okay, can A donate blood to B? What do you think? Can A donate blood to B? No. Okay, A cannot donate blood to B because the antigens are missing. Immediately, there is going to be an enzyme reaction. Okay, meaning there's going to be a reaction, adverse reaction. Can A donate blood to AB? What do you think? The answer is yes. Why? But then you'll be like, sir, but AB hai. See, A can donate blood to AB because AB has both the antigens. Even if you take A and you give it to AB, it is still going to take it. You take B and you give it to AB, still it is going to take it. So yes, A can donate blood to AB. Can A donate blood to O? What do you think? The answer is no. Okay, because O has no antigen, immediately it is going to detect it as foreign. Let's try to see. Now B. Can B donate blood to A? Answer, no. Can B donate blood to B? Yes. Can B donate blood to AB? Yes. Because once again, AB has both the antigens. And can B donate blood to O? No. Okay. Let's try to see next thing. Okay. Okay. Now the next one is your AB. Okay. Can AB donate blood to A? What do you think? No. See, A can donate blood to AB because AB has both the antigens. But if AB is giving blood to A has only one antigen. The B antigen, which is going to be extra, is going to cause a problem. So, guys, no. AB cannot give blood to A. Can AB give blood to B? No. Once again, for B, A is going to be extra. Can AB give blood to AB? Yes, AB can give blood to AB. And now, can AB give blood to O? No. Okay, immediately both of them will be detected as foreign. So no. Okay, and now lastly, guys, lastly we have O. So see, can O give blood to A? Yes. Okay, because there is no antigen on top. So yes. Can O give blood to B? Yes. Can O give blood to AB? Yes. Can O give blood to O? Yes. So see, all of them, they are going to be yeses out over here. So what is happening? So here we are going to see, guys, this thing. Okay, so we are seeing all four ticks out over here. So because we are having all four ticks out over here, guys, what is this? This is your universal donor. This becomes your universal donor. But now, if you look out over here, okay, you have AB. So AB is going to have all the ticks out over here. Yes. But who were these? These were your acceptors. So what is this AB? AB is your universal acceptor. Okay, so you can say this is your universal acceptor, or you can also say this is your universal recipient. Okay, universal acceptor or universal recipient. Everybody all right with this thing, guys? Sure, understood this thing. But only O negative can be universal donor other than O positive. No, no, no. See, we have not got into your RH positive and RH negative. What you're saying is right. But for the time being, we are into this thing. Then slowly we are going to go into your next part when we'll add on to your positive negative. Okay, everybody fine with this thing? So for the time being, we are going to call this thing as universal donor and universal recipient. Everybody okay? Fine. Chalo. But now, guys, what did I tell you? Afterwards, now see, when was this thing happening? See, this was happening in 1902. Okay, in 1902, all this thing was happening. But now, guys, you know what? What happened was later on, when people were doing blood transfusions and all, they found out that even when the two blood groups were same, like we had A positive and I mean two A blood groups or two B blood groups, if they were being transfused, then also people started having some various reactions. So slowly, what happened was, they tried to find out that what is the problem. Like, see, technically, A blood group, when you give it to A blood group, it should not have an issue, but still people were having issues. So now what happened was the same guy, Carl Landsteiner, okay, the same guy, he was working on your blood groups once again. And now the year is 1940. At that time, Karl Landsteiner and his colleague Wiener, they found out that, oh, instead of A, B, and O, there is one more antigen which is present on the top of your RBCs, and that is nothing but your Rh antigen. So the next one, we have Rh antigen. Guys, am I lagging by any chance? Hello, can you guys hear me properly? Okay, okay. Okay, the one who is lagging 1 minute or 1 minute. Devang, if you can just log out and log in again. Anybody who's having, lagging issues, just log out and log in again quickly. Just a moment, guys. We'll just wait for him to log in. Okay, he's back. Okay, so now, guys, they found out that this is another antigen. Here is, guys, 1940. And who were the people? Once again, your Karl Landsteiner. And this time, he was with another guy whose name was Wiener. So Karl Landsteiner and Wiener. Okay, so what did they found out? They found out that we have one more antigen which is present, which is called as the Rh antigen. And what is this Rh antigen, guys? This is actually your rhesus antigen. And where does this rhesus antigen come from? This is the one which comes from the rhesus monkey. Okay, it comes from the rhesus monkey, and because we are from the lineage of rhesus monkey, we see this thing on our RBCs, and this was the one which was actually creating problems for us. Okay, but how is it creating problem? Let's try to see, guys. Okay, so now let me just see. So guys, now what happened was, see, initially we had only your four blood groups like this, right? A, B, AB, and O. But now, because there was addition of your third antigen, this entire thing now just got changed into 8 blood groups. How come? Let's try to see. So guys, I told you there is going to be one more antigen on the top. So that antigen is your Rh antigen. So there can be two possibilities. If Rh is present or if Rh is absent. Okay. So let's say on the first line, Rh is going to be present. So what are we going to have, guys? So see, you are going to have A, Rh. So here B, Rh, AB, Rh, and O, Rh, right? So now, guys, now what happens? So basically, the same blood groups are going to be divided into two types, meaning A can be either with Rh or A can be without Rh. B can be with Rh, without Rh, with Rh, without Rh, with Rh, without Rh, right? So now let's try to see what do we call this blood group. So guys, actually, what is this? This blood group is called as ARH positive. This is BRH positive, ABRH positive, ORH positive. But now, guys, with love, what are we going to say? We just call ourselves A positive or B positive or AB positive or O positive. This way, this is how we denote ourselves. Okay, but now let's try to see on the other side. So guys, here RH antigens are not present. So what is this going to be? This becomes ARH negative, BRH negative, ABRH negative, ORH negative. And now guys, what is this going to be? With love, we call this thing as A negative, B negative, AB negative, and O negative. This way. This is how it is going to happen. But now, guys, now let's try to see. Let's go back out over here. See, guys, when we talked about your blood group O, we said blood group O was universal donor. Why? Because it did not have any antigen on the top. Because it did not have any antigen on the top, it did not bring about any reaction in the other recipients, right? That is why it was universal donor. And we said we have AB as universal acceptor. Why AB is universal acceptor? Because it has all the antigens present on top. So no matter whatever you throw at him, he is going to take it, right? Now using the same logic out over here, guys. Now, who is going to be the real universal donor, guys? Just use the same logic. Guys, what do you think? Who is the real universal donor? Quickly, quickly, quickly. Very nice touch. Very nice, Devan. So what is it? Guys, see, using the same logic, we are going to have O negative. O negative, he is the real universal donor. Why? Because there is no antigen present on top. Okay, real universal donor. No antigen present on top. And now, who is going to be the real universal acceptor, guys? Quickly. No, 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 no. Real universal acceptor. Very nice, Ansh. Yes, very nice, Sneha. Okay, what is this? This is going to be AB positive. Why AB positive? Because see, it is going to have all the antigens present on top. Whether you throw A positive, whether you throw B positive, whatever you throw, it is going to take it because it has all the antigens. So it can take from all of them, all the positive ones also, and all the negative ones as well. So guys, AB positive, this is the real universal acceptor. Okay, is everybody okay with this thing, guys? Sure, no problem. Okay, okay. Is everybody clear, guys? Guys, once again, if you want, you can just have a look at both these things. Okay, just have a look once. If you have any doubts, quickly ask. Okay, now the next thing that we're going to see is, guys, sometimes, there are going to be some issues when you have, like, different types of parents with different blood groups. So we will just see that how it is going to work. Okay, so which blood group is, like, going to create a problem, which is not going to create a problem. Let's try to see how it is going to work. So now let's see this thing, guys. Okay. So the next thing, so okay, before that, let me just see, guys. I told you this is your ABO blood grouping system, but are there any other blood grouping systems that are available? Yes. Okay, there are other blood grouping systems also. Okay, for example, okay, so for example, guys, we are going to have Duffy. Okay, these are other ways in which your blood groups can be classified. But then your ABO blood grouping system is the most widely used. So we have Duffy, we have Kid, we have MNS, we have Lewis, okay, etc., etc., etc. These are other blood grouping systems. Okay, but now guys, we have this problem which is called as your erythroblastosis fetalis. Erythroblastosis fetalis. Okay, or this is also known as hemolytic disease of the newborn. Okay, hemolytic disease of the newborn. Okay, if you want, you can just call this thing as HDN. Okay, but now guys, what is going to happen? What is the problem? Let's try to see. So guys, let's say we have a male. Okay, we have a male and we have a female. Let's say they are about to marry one another. But this male, he is going to be Rh positive. Okay, and female, let's imagine she is Rh negative. Okay. Now, guys, see, for every gene, you're going to have, like, for every character, you're going to have two genes. So let's imagine this male, he is Rh positive, Rh positive. And let's say this female, she is homozygous for Rh negative. So she is Rh negative, Rh negative. Let's imagine. Now, guys, both of them, they have decided to marry and they are going to have a baby. So now what will happen? See, this one is going to be Rh positive. This one is going to be Rh negative. The gametes are going to be this way. But at the time of fertilization, what are we going to get? You are going to see Rh positive, Rh negative. So guys, what is the baby going to be? In this case, the baby is always going to be Rh positive, right? Now, let's try to see how is the situation. So guys, let's imagine this is that female. Okay, so let's imagine we have this female, okay, who is bearing the baby, something like this. Okay, so now let's assume this situation. So let's say the baby is out over here in her womb, like this. Now, what is the female's blood group, guys? See, the female's blood group, she is Rh negative. But let's have a look at the baby. What is the baby? The baby is Rh positive. Now, can this be possible that one part of your body is having positive blood group, one part is having negative blood group? एक हाथ में positive है, एक हाथ में negative. No, it is not possible at all. It cannot happen that way. So, guys, now is this going to be a right situation? No. Now let's try to see. Guys, if by any chance this Rh negative comes to know that, oh, there is this Rh positive antigen present out over there, what is this Rh negative going to do? Immediately he will start producing Rh antibodies, like, antibodies against your Rh positive. These Rh antibodies, they are going to come and, yeah, kachak, they are going to kill this baby right in the womb itself. What is this? This is nothing but your erythroblastosis fetalis. But then, guys, then does it mean that, oh, the one who is going to be Rh positive cannot marry one who is Rh negative? Is it right? No. Okay, so otherwise, from tomorrow onwards, you will have to hold a placard, and wherever you're going, hey, Rh positive, Rh positive, Rh positive, Rh positive, Rh positive. Okay, are we really going to do that? No, right? And what about, like, if you fall in love with other person, love is blind. Already we are blind. You fall in love, you're blind. Now you're not going to look out for all these things, right? Now let's try to see what happens, guys. Okay, now, sorry. Okay, so for the first time, guys, when they are going to mate with one another and when they are going to have a baby, the baby is going to be Rh positive. Mother is still going to be Rh negative. But guys, the mother is never going to come to know that the baby is Rh positive. Why? Because we are going to have placenta out over here. And guys, this placenta, it prevents the entry of your blood, meaning it prevents the mixing of the mother's blood and the baby's blood. right? So this placenta will never let the mother know that, oh, in her womb, there is some Rh positive antigen. Okay. But guys, this is for the first time. Okay. Now, what happens, guys? So, first, the baby is going to deliver. I mean, the baby is going to grow. And at the time of the first delivery, this is still going to be fine. Like, the baby is going to be delivered and all. But you know what, guys? At the first time, when the baby is going to be delivered, the placenta is going to rupture. And when the placenta ruptures, the mother's blood and the fetus's blood, they can mix with one another. And now what? If they mix with one another, immediately the mother comes to know that, oh, there is something called as your Rh positive antigen. Immediately she starts producing antibodies. Now, when these people are going to try for a second baby, now mother already knows that there is antigen, right? Now what happens? She starts producing antibodies. But guys, what did I tell you? Antibodies can cross the placenta. So when they will have the second baby, now the mother will produce antibodies. These antibodies will now cross through the placenta and they will kill the baby, right? This is erythroblastosis fetalis. Okay. But now, so does it mean that these people who are like Rh positive, Rh negative combination, they should have only one baby only? No, it is not so. Why? Because you know what, guys? Okay, 1 minute. Let me just whatever I had written. So second delivery. Okay, if nothing is done, then at the time of second delivery, basically the fetus is killed. The second time, the fetus, this is killed. Okay, but now let's try to understand, guys. What do we do? Okay, to avoid this situation after the first delivery. Okay, after the first delivery, guys, we are going to give the mother an injection. Okay, and what is this injection, guys? This injection contains anti-Rh antibodies. Okay, so this is going to contain anti-Rh antibodies. Okay, so now what is going to happen when these anti-Rh antibodies they are given? Okay, or you just call this as Rh antibodies. Okay, so either your Rh antibodies or anti-Rh antibodies. So when you give this injection to the mother, now what happens? If by any chance, okay, after the first delivery, let's say some blood cells had mixed out over here, these antibodies, preformed antibodies, they are going to go and they are going to destroy these Rh antigen. Now, if they have already destroyed the Rh antigen, guys, will the mother ever come to know that there was this Rh antigen present in the body? No, mother will never come to know. And if the mother never knows, will she ever produce any antibodies? No, she will never have any antibodies. And if there are no antibodies, the mother is not sensitized. Immediately for the next time, they can have a normal baby once again because the mother never produces antibodies. Everybody okay with this thing? Got this part? Okay, so always remember, if you have any situation like this where mother is like negative, you are positive, in order to avoid the situation after the first delivery, you have to take this injection. Okay, this injection costs around 2,500 in the current circumstances. Okay, fine. Okay, chalo. So this is what we need to know with regards to the entire first half of the chapter, where we talked about the entire immune system. We talked about what are the parts of the immune system, what are the cells involved, what are the different aspects of the immune system and all. So this thing. The next part of the chapter, yes. Most of the time, see, they will always check your blood groups. Okay, and then only you'll have to take it. Sometimes when the blood groups are matching, let's say your mother is positive, father is also positive, then there is no need for taking this this injection. So before before the delivery, the mother's blood group is going to be checked. Okay, mother's blood group will be checked, and then they will decide whether we have to give this injection or not. Now someone is asking, what if it is the other way around? So very simple. See, if it is the other way around, there is no issues at all. So let's imagine male is Rh negative. Okay, so he is going to be Rh negative, and let's say the female is Rh positive. So who is dominant? So guys, Rh positive is still going to be dominant, right? So let's say when they are going to have a baby, their baby is going to be Rh positive, Rh negative. So ultimately he is going to be Rh positive. So he is going to be Rh positive, right? So mother is also Rh positive. So mother is, let's imagine she is also Rh positive, and the baby is also Rh positive. Hey, hello, 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 guys. Can you hear me? Somebody signed me out. Hello, can you hear me, guys? Hello, hello, hello. Okay, 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 okay. Someone has logged into the account, so that is why, I mean, I got logged out of the meeting. Okay, so see, guys, here mother is also Rh positive, fetus is also Rh positive. Is there going to be any problem? No, there is no problem. So if the cases is reversed, there is no issue. The problem only arises when you have different blood groups, Rh positive, Rh negative. Same way you can have it with your AB positive, AB negative, or B positive, B negative. Okay, then also the same kind of thing. So when there is this positive negative combination, that time you'll have this problem. Okay, understood this part, guys? Okay, so now, guys, the next part. Okay, so we're done with the first of the chapter. Next part of the chapter is going to talk about all the different kinds of diseases that we are going to encounter. Like, you're going to talk about AIDS, you're going to talk about your cancer. We have like typhoid, pneumonia, we have ascariasis, malaria, all these different kinds of diseases that we'll talk about. Okay, but then all that part will start off in the next class. Okay, so once again, guys, we'll stop over here for the day and I'll see you in the next class. Till then, take care. Bye bye, everybody. Bye bye. Happy Independence Day. Enjoy your day, guys.